Hello guys, I am Shubham from ShubhVed Tutorials. This video is about quantitative traits. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. See, first of all, when we start studying genetics, okay, there is Mendel laws, right? Okay, right? And we have studied previously, like there are two parts, okay, Mendelian inheritance and non-Mendelian inheritance. Alright, so we can say like that this video is about quantitative traits, but Mendelian inheritance, the Mendel laws, are followed by qualitative traits. Okay, qualitative traits and non-Mendelian inheritance can be considered as quantitative traits. Quantitative traits. Okay, because these quantitative traits do not follow Mendelian inheritance or Mendelian laws. Right. So these traits are non-Mendelian. Right. And if they are non-Mendelian, means there is nothing like dominance and recessive. Okay. In this quantitative alleles, there is no dominance and recessive. We can study quantitative traits on the basis of qualitative traits. So we will compare those. First point can be written as qualitative traits follow Mendel. Okay, this don't follow Mendel. Okay, Mendelian inheritance, or we can say Mendelian laws. Then second point can be written as, here it will come continuous variation in phenotypes. And here there are no variations, means discontinuous variation. Discontinuous variations, or we can say discrete phenotypes are seen. Okay. Then polygenic inheritance, and here single gene. Okay, right? Then polygenic means there are many genes. In Mendelian means in qualitative traits there is only single gene we consider. Okay, and they are alleles. Right, here there is nothing like allele, and if there is there, the next important point is contributing and non-contributing alleles. Okay, and here there is dominance and recessive alleles. Right? And one more important point is, in qualitative there is like allelic genes, means there are alleles according to previous point, and here non-allelic genes. And here we can see that phenotype in qualitative, phenotype is equal to what phenotype we see, that is dependent on genotype. But here phenotype is not only dependent on genotype, but also environmental factors. Okay, and this is mostly a population based study, quantitative, metric traits, we can say that. Alright, quantitative can also be said metric traits. Okay, so here it will be traits, quantitative traits and qualitative traits. Right, so this is the just the intro part. Now with the example we will see what are those actually. First of all, qualitative traits we will see. Qualitative traits, just said this is a Mendelian. Okay, Mendelian inheritance. Right, so see, all know this cross, right? Mendel, parental, this is true breeding parents, this is F1, and here F2. F2 is what? Cross, right? F2. Okay, we cross with F1 and bring about 3 are tall and 1 dwarf. Right? Means see, in the F1 this is tall and this is dwarf. So all F1s are tall, right? Because of this dominance gene, right? Capital T. These are tall. Okay, but in quantitative these things are not matter, right? So here we know in this qualitative, this is first of all these two homozygous is tall. Okay, and this heterozygous is also tall due to dominance allele. Right, now we will see quantitative traits with example. Okay, see, these are the true breeding, right? Parental generations. Here F1 will be this. Okay, but we know this genotypically, but phenotypically, say, suppose in this example is wheat kernel color. This is the example. Okay, so suppose this is red and this is white, true breeding. This will be intermediate red. Intermediate red. Got it? Means we can say that it is like, okay, like partial dominance, or we can say incomplete dominance. Okay, but here we are talking the word about dominance. Here is nothing like dominance and recessive alleles. Okay, here the F1 will be intermediate. Okay, in this things also we saw intermediate, partial dominance and incomplete dominance. So it can be correlated with that. But here in quantitative traits, the capital ABC, okay, the capital ABC are contributing alleles. Contributing alleles. And small ABC are non-contributing alleles. Okay, so that is the case here. So this is a chart through which we will see what is quantitative traits in detail much. So this is a dark red, see, and this is a white, right? Intermediates are intermediate red, capital A, small b, means this is the phenotype as we saw here. I will again write here for more easy. We can say, right, suppose this are the two breeding, right, and this is the inter for F1, one capital one small. This is the P generation, This is the F1, right? And F2, we know F2 is obtained by the cross between F1. So here we can see when for the F2, okay, F2 we get this type of phenotype. Okay, so dark red is 1, right? Dark red is 1, then moderate red is 6, red is 25, intermediate red is 20, okay, light red is 15 again, very light red is 6, and white is 1. Okay, so these are the things. Now number of contributing alleles, right? Contributing alleles here, all the 6 are the contributing alleles. Means for the first one, dark red. So if I say like that, the dark red, how 6, basically you can say like that, A 1 2 3 4 5 and 6. So all these are responsible for producing the dark red color, right? And moderate red will come up with only 5 alleles. Okay, means we can say A, B, B, C and here C. This can be moderate red. Okay, so when we will plot this, then we will get this type of curve. Okay, bell shaped curve, right? Majority are intermediate red. We can see in the chart 20 are intermediate red. See 20, right? So what we can see here in population, the intermediate phenotype is the most number is the intermediate red, and this are the extreme phenotype. Okay, so what we can say here, so this are known as extremes, right? Extreme point, and from extreme point it goes on increasing and again goes down decreasing. See, okay, so here this is known as the intermediate point, it is the highest. So this is a quantitative trait, this we want to understand. Okay, so here we get a bell shaped curve, bell shaped curve. Okay, means like this, right? Here will be the intermediate and here will be the extremes, and it will go on increasing like this. Okay, so these are the variations we see, continuous variations. These are known as continuous variations from here, right? Continuous variations, thus it is known as quantitative traits. So it is seen that environmental factors are also responsible, or we can say contributing alleles and non-contributing alleles. These terms are to be considered when talking on quantitative traits. Okay, so now the calculation things of quantitative traits. Calculation is basically we can find out the number of gene pairs, okay, or we can say number of genes, right? So extreme phenotype we will consider. Extreme phenotype, as we saw here, the extreme phenotype is dark red. It is having ratio 1 and total is 64, right? So that ratio we want to keep in mind, that number ratio 1/64. Okay, extreme ratio, right? Extreme phenotypic ratio, I will write here extreme phenotypic ratio. So this is the extreme, that is dark red, 1/64. Okay, so 1/64 is what? 4:3. So this 3 is an, means 3 genes are involved for this trait. Okay, number of gene pairs it is better to write gene pairs here. Gene pairs. Okay. Now from the gene pair we can calculate phenotypic classes, means how much phenotypic classes we can see in this quantitative traits. The formula for this is, I will write here. So number of phenotypic classes. Okay, so here formula is 2n plus 1. Okay, so n is 3. So here comes 7. So 7 are the phenotypic classes, right? Let's see. This is the thing. Correct? Yes. See, we can see here. See, this is 1, this is 2, this is 3, this is 4, this is 5, this is 6, and this is 7. So 7 phenotypic classes we are seeing. So this is the calculation part. Also, that is how to analyze this quantitative trait. Okay, how can we understand? This is a concept known as quantitative trait locus analysis. Okay, quantitative trait locus means QTL. Okay, alright. So the regions within the genome responsible for this quantitative trait are known as quantitative trait locus. Suppose I draw here a chromosome, right, and this is a gene segment for quantitative trait. So this is known as quantitative trait locus. Now how to do analysis of this? So we can do analysis by phenotypic data and genotypic data. Right, so phenotypic data, we take different organisms, right, differing in genotype but having the same traits. Okay, the genotype is different but having the same traits. And genotypic data, we use the DNA markers. Okay, so I will write here about genotypic data. DNA markers we use. Okay, so DNA markers used are basically SNP, single nucleotide polymorphism, SSR, simple sequence repeats, right, then RFLP, restriction fragment length polymorphism. So how the analysis is performed? Basically with the help of what we can say, same like we perform crosses. The parentals are crossed to get the F1 and then F1s are crossed to get F2. Okay, then the data, genotypic and phenotypic data are removed, and then using that we can analyze the markers, means the linked markers will get segregate according to the phenotype. Okay, and the unlinked markers will not show any segregation or any correlation with the phenotypes, right? So that like this analysis is done. So we want to remember this: DNA markers, SNP, single nucleotide polymorphism, SSR, simple sequence repeats or microsatellites also known, and RFLP, restriction fragment length polymorphism. So this is the method of analysis. Okay, so that's it for this video. I hope this video helps you. If this video helps you, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever new video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.